Thanks everybody for joining us. Sorry about that little delay at the beginning there. This is the May 2025 Seminar on Physical Genomics. My name is Benjamin Keene here at the Center for Physical Genomics and Engineering at Northwestern University. We're very happy to welcome our invited speaker today, Professor Kristy Stengel. Dr. Stengel is an assistant professor in the Department of Cell Biology at the Albert Einstein College of Medicine. A little background, she completed her PhD in cell and molecular biology at the University of Cincinnati and Cincinnati Children's Medical Center. After obtaining her PhD, she pursued postdoctoral studies with Scott Hibert at Vanderbilt University. In 2021, she joined the faculty at Albert Einstein as an assistant professor, where she is continuing her work using cutting-edge technologies to define the mechanism of action of oncogenic transcription factors. Professor, thank you so much for sharing your work with us today. A quick note for our audience, we will have a Q&A session at the conclusion of the talk. So if you want to, you can just type your questions into the Zoom chat, really anytime you think of them, and we'll address them at the end. If you're interested in the research and training programs going on here at CPGE at Northwestern, you can visit us at physicalgenomics.northwestern.edu, or you can check out our archive of seminars and talks on our YouTube channel. And we're also on Twitter, X, and Blue Sky if you want to follow us there. So that's my spiel at this point. Professor, I think we can turn the floor over to you. Thank you so much for joining us again.
All right, great. Thanks for having me. So everyone can see the slides okay and hear me okay?
That's perfect, yes.
Okay, great. Well, thanks again for the invitation and thanks for the introduction. I'm really excited to be here to share some of my group's work on understanding sequence-specific transcription factors. Let me just minimize this really quickly. Great. Okay, so I teach I co-lead a class on gene expression here at Einstein, and I give a lecture on transcription factors, and this is always one of the first slides I show. And I always, my lab is particularly interested in transcription factors that regulate blood development. And when you stop and pause and you realize that in spite of the fact that in this hierarchical system where all of these cells essentially have the exact same copy of DNA, there's tremendous phenotypic and functional diversity. So an hematopoietic stem cell gives rise to cells that are capable of carrying oxygen throughout your body, of producing platelets to allow your blood to clot, producing antibodies that help fight infection. And again, all of these cell types, by and large, have the exact same genomic information. And so what accounts for this tremendous functional heterogeneity is, of course, which genes are expressed. So how and when specific gene programs are initiated and maintained is really driven by a class of proteins called transcription factors. And I like to think of these as really the master regulators of cell identity and function. And it's estimated that the human genome encodes roughly 1,600 different transcription factors. So that means that the human genome uses 8% of the genes to encode for proteins that regulate when and where other genes are expressed. And I think this really underscores the importance of these proteins for normal cell development and function. What also underscores the importance of these proteins for normal cell development is the sheer frequency with which they're altered in diseases like cancer. And so my group is particularly interested in myeloid malignancies. And this is just a summary of some of the most frequent mutations that you see in acute myeloid leukemia. Let's see if we can get a laser pointer. And so what you can see is about 22% of these cancers have mutations in a myeloid transcription factor. Almost another 20% have common translocations that involve transcription factors and lead to transcription factor fusion proteins. And even what's classified as tumor suppressors are in fact genes that encode proteins that regulate gene expression. And so not only does the frequent co-occurrence of these mutations in transcription factors underscore their importance in cancer biology, we also know from studies like the cancer dependency map that transcription factors are profound vulnerability in these cancers. However, by and large, we've done a poor job capitalizing on that vulnerability because factors are also notorious for being undruggable. But we do know for transcription factors in which there are existing therapies, typically ligand binding transcription factors like the estrogen receptor, androgen receptor, those therapies really form the cornerstone of therapeutic intervention. And so I think for these more undruggable transcription factors, the more we understand how they function, the greater our likelihood of being able to intervene therapeutically. So one of the reasons that I hope I'll be able to convince you that we really don't understand enough about how transcription factors work is because oftentimes if you read a textbook, you'll see something like this, a cartoon like this, a transcription factor sitting on DNA at its target gene, and that's how it regulates gene expression. And my group and many others use these genome-wide approaches, and you'll see these nice peaks where your transcription factor is binding. And I think these depictions of transcription factors really give this idea that these are very static molecules, but really that couldn't be further from the truth. And so if you take approaches that allow you to look and actually watch these transcription factors move around in living cells. So here we've added a halo tag to our transcription factor of interest, RUNX1, and this allows us to watch RUNX1 move around and actually associate with DNA in living cells. And this video was obtained by our collaborators, John Hobbs and Robert Coleman. And so when you watch RUNX1 in living cells using this approach, you can see kind of a haze of gray, which is the freely diffusing RUNX molecules. And when it binds to DNA, it slows down enough that you see this distinct focus of light. And they can quantify the off rate of those binding events. And what they observe is the vast majority of RUNX1 binding is what they classify as unstable, roughly 80%. And those events last for less than a second. But even the most stable RUNX1 binding events, which is roughly 20% of those binding events, only persist for 10 to 15 seconds. And so I just use this as an illustration to make the point that transcription and transcription factors are highly dynamic. And transcriptional regulation has to be highly nimble and dynamic in order for the cells to respond to a variety of stresses or signaling cues. Okay, so the question then becomes, how do we study proteins that are this dynamic in nature? And we know that so we think the kinetics of the experimental systems that we're using really do matter. And we know that the most rapid way to inactivate a protein is through small molecules. But as I mentioned, most transcription factors are notoriously undruggable. And so historically, we haven't had those types of tools at our disposal. And so really how we've come to understand many of the transcription factors, particularly the lineage factors that are responsible for cell identity, is through genetic means. So either mouse models, genetic knockouts, or even shRNA or now CRISPR. The problem with these approaches is that they're very slow to reduce the protein level. And if that transcription factor has other targets that are themselves transcription factors or signaling molecules that will ultimately impinge on gene expression programs, by the time you're able to assay changes in gene expression, typically through an assay like RNA-seq, what you have is this accumulation of both the direct effects of inactivation of your transcription factor and gene expression, but also the secondary, tertiary, compensatory changes in gene expression. And so while animal models in particular have been fabulous in allowing us to understand what the phenotypic implications are for loss of a transcription factor, almost the gene expression assays that are done are almost a reflection of those phenotypic changes rather than the direct transcriptional mechanisms that actually led to those phenotypic changes. And I think a great example of what a small molecule can really do with the resolution of changes in gene expression, I like to show this example. This was generated by one of my friends during my postdoc, where if you have a small molecule that targets a transcriptional protein, JQ1 is a famous small molecule, it's a BET inhibitor that triggers pausing of RNA polymerase. It prevents pause release into productive elongation. And all you need to know is this is a mapping of transcriptionally engaged polymerase as it travels across this KIT gene. And within 15 minutes of treating the cells with JQ1, you see induction of the pause. And the polymerase that had passed that pause, you can actually watch it as it exits the locus. And by 30 minutes of BET inhibition, this gene is maximally silenced. So within 30 minutes, you've basically had the direct effects of using the small molecule of on gene expression manifest. And the longer you go out beyond that just 30 minutes, you risk the accumulation of, again, secondary and compensatory changes in gene expression. And so this has really limited our ability to first identify the direct targets of sequence-specific transcription factors, which I hope I'll be able to convince you throughout this talk is necessary for our ability to understand how they regulate gene expression. Okay, so again, you can think of inactivation of a transcription factor as dropping a stone on a pond. You'll initially have a first wave of changes in gene expression. These are likely the direct effects, but then those will give rise to secondary and tertiary waves. And ultimately, the surface of that water is changed in a process that we think of as transcriptional adaptation. That's just this accumulation of, again, direct effects on gene expression, but also compensatory changes and leading to a new transcriptional state. And so what we want to do is compress this time frame for studying inactivation of transcription factors from days to weeks, like we have in these genetic systems, to really the order of minutes to hours. And the way that we've been doing this is through the use of Degron technology. So we incorporate a Degron tag into an endogenous transcription factor locus. Now, when that transcription factor is made, it has this extra peptide on the end that renders it sensitive to a PROTAC called DTAG. So this is a heterobifunctional molecule. One side binds to that FKBP tag, the second side binds to an E3 ubiquitin ligase, and by inducing ternary complex formation, you get the rapid ubiquitination and subsequent degradation of your protein of interest. And now we can achieve degradation of these proteins within minutes to hours. Okay, so I just want to talk briefly about the transcription factor that I'm going to discuss today. This is a transcription factor, RUNX1, that's critical for regulating gene expression programs in hematopoiesis and other tissues, but we're focused on hematopoiesis. So in animal models, we know that if you inactivate RUNX1, it's embryonic lethal. You don't get definitive hematopoiesis, and the embryos have the severe hemorrhaging in the CNS. So they don't really have platelets that form, and they don't develop definitive HSCs. In addition, using conditional models, others have seen that if you bypass this requirement for RUNX1 during embryogenesis, you do in fact get viable animals, but they have, depending on the cell type in which it's deleted, a variety of effects throughout the hematopoietic system. Now, in addition to this important role in development, RUNX1 is one of the most frequently altered genes in myeloid malignancy, and this happens in a couple of ways. So RUNX1 is the target of the 821 chromosomal translocation. So this translocation results in the fusion of the DNA binding domain of RUNX1, also called AML1, to this co-repressor molecule called EDO. And this is the most frequent translocation in AML. But in addition to this translocation, there's also frequent loss of function mutations that occur in RUNX. These tend to be frameshift mutations that lead to loss of the protein, or missense mutations tend to accumulate in the DNA binding domain of RUNX1. These are associated with somatic MDS or AML, but there's also rare families that have germline mutations in RUNX1 that leads to a condition called RUNX1 familial platelet disorder. So these individuals have problems with blood clotting, and they're also predisposed to the development of myeloid malignancy at an early age. Okay, so my talk today, the first part of my talk is all published work. It's going to be just some highlights from the first attempt we made to use this Degron technology to understand how an oncogenic transcription factor worked. And that involved understanding what I'll refer to as AML1 EDO, but keep in mind AML1 is just another name for RUNX1. And then I want to use that first part to introduce some concepts that we've observed using this Degron technology, and then further address those using the example of the wild type RUNX1 protein in the second part of my talk. Okay, so the 821 translocation, RUNX1 or AML1 is typically thought of to function primarily in gene activation. There's clear evidence that it can both activate and repress, but it's predominantly thought of as an activator of gene expression programs that are critical for normal hematopoiesis. EDO, the translocation partner, is really functions as a scaffold. So it forms these heterotetramers with other family members, and they both bind to sequence-specific transcription factors, and they recruit co-repressor molecules, including histone deacetylases, to turn off gene expression. And so the consequence of that translocation is really the constitutive recruitment of these HDAC complexes and other repressor complexes to RUNX1 target genes, leading to their constitutive repression. And it's thought that this is really what induces leukemogenesis. So there's a lot of beautiful biochemical work that has been done, many years ago now, really demonstrating that the mechanism of gene repression is, or the fusion protein actually functions through eliciting gene repression through the recruitment of these co-repressor molecules to RUNX1 gene targets. Now, in spite of this kind of historical biochemical data, around the time we first initiated these studies, there were a lot of papers that were beginning to be published that suggested that this AML1 EDO protein actually didn't only function as a repressor molecule. It could actually activate a lot of gene targets. Many of these were canonical oncogenes, and it was beginning to be proposed that AML1 EDO may actually induce leukemogenesis through activation of oncogenic genes, in addition to simply repressing normal RUNX1 target genes. And so we set out with a very, very simple question in mind, which was, can we use this Degron technology to define what are the direct transcriptional targets of AML1 EDO? Are these activated targets really directly regulated by the fusion protein? Are these perhaps secondary events? So we use CRISPR-based engineering to add this FKBP tag at the 3' end of the RUNX1 EDO locus. We also, in addition to incorporating the Degron tag, we incorporated an HA tag that allowed us to do a number of biochemical assays. And then there's a P2A ribosomal skipping sequence and a fluorescent marker that allows us to use flow sorting to select for the cells in which we get successful integration of this cassette. So the only way that M-cherry in this case becomes expressed is if this Degron cassette is integrated in frame into a transcribed open reading frame. And when we do this, we see that in this 821 cell line, following CRISPR engineering, we can detect the HA tag on AML1 EDO, and AML1 EDO migrates more slowly on the gel, consistent with the addition of the tag. So this now gives us a system where we can treat with this DTAG molecule and degrade AML1 EDO. Now, I will say, this was done, several years ago now. The chemistry around these DTAG molecules has gotten even a little bit better. This was before these were even commercially available. And so this degradation is actually pretty slow, but still at the time was a vast improvement over the days in which AE had been knocked out previously and then looked at gene expression. So within 2 to 6 hours, we were eliminating the protein. Okay, so not only do we need a system that will allow the kinetic resolution of protein elimination to allow us to monitor changes in gene expression, we need a direct assay for measuring changes in transcription. And so the technique that we use is PROTAC. It's essentially a nuclear run-on in the presence of biotinylated NTPs. So as the polymerase transcribes, if it incorporates one of these biotinylated NTPs, it's chain terminating. So then you can purify just the nascent transcripts based on the biotin-streptavidin interaction. And if you sequence from the 3' end, you'll know exactly where the transcriptionally engaged polymerase was transcribing at that time. I just want to quickly run over some of the features of PROTAC that I'll discuss in more detail as we go through the talk that we find particularly useful for quantifying changes in gene expression. The first is that you're going to get stranded information. So at promoters as well as enhancers, transcription actually initiates bidirectionally, but you'll only get sustained elongation on the sense strand. Okay, so here you have the coding transcript shown in red and this anti-sense transcription shown in blue. What's very useful for us is that you will also get non-coding transcription, and this includes enhancer RNAs. So we can, in one assay, we can measure changes in gene transcription as well as changes in enhancer activity, because it's been shown that changes in eRNA synthesis actually better correlates with enhancer activity than even histone marks that we commonly associate with enhancers like H3K27 acetylation. And then finally, this won't be relevant for this talk, but it gives further mechanistic insight because PROSeq is actually able to detect both the paused as well as elongating polymerase. And so you can quantify changes in polymerase pausing using this assay. Okay, so here's an example of a gene that we found to be regulated by the AML1-EDO fusion protein. You can see in the absence of the tag parental cell lines, it's not transcribed at all. Before addition of the DTAG molecule, it's not transcribed at all, but within 1 hour of adding this DTAG to the cells, you can start to see polymerase showing up through the gene body, and that polymerase continues to accumulate through 6 hours. When we looked globally at the genes that changed, we were really surprised to see that the number of genes directly regulated by this fusion protein were actually quite small compared to what had been previously reported. So if you look for genes where there was an increase in gene body transcription following degradation of the fusion protein, so that would indicate that these genes, the fusion protein was maintaining them in a repressed state. You can see at 1 hour, 13 genes that come on, they remain on at 2 hours, where you get an accumulation of another 46, 50 or so genes, and those remain up at 6 hours. So you get a derepression that's relatively stable, and those genes stayed on for the duration of the time course. In contrast, looking at genes that showed a decrease in gene expression that might be consistent with this fusion protein activating genes, they were highly variable. So many of the genes that would be decreased at one time point didn't stay decreased at another. And if you look at just the 7 that were shared amongst time points, by the time you get to the 6 hour time point, they've actually started to recover. And here with the arrows, I've highlighted some of the direct targets of this fusion protein that are themselves transcriptional regulators, which again highlights why we think we need to look very early following removal of a transcription factor to really identify those direct gene targets. And I think this also further illustrates this. So if we look longer after AML1-EDO removal, out through 24 hours, and now we're looking by RNA-seq as opposed to this nascent transcript profiling by PROSeq, what you see are these waves of transcriptional changes that happen. And if you just quantify the number of genes that come off and go on, yes, you might look at this and say, well, I can't tell if this fusion protein is really activating or repressing gene expression. And by 24 hours, in fact, there's more genes that are turned off than turned on. But if you compare that to the nascent transcript profiling over the very short time courses, what you see is overwhelmingly, there's more genes that are upregulated upon AML1-EDO degradation, consistent with this role in transcriptional repression. So, you know, historically, the way we've tried to get around this, we've known that when we use these genetic models, there's going to be waves of transcriptional changes, and some of them are going to be direct and some of them are going to be indirect. But we always thought that the way we could identify the ones that are direct is by doing something like ChIP-seq or cut and run, where we actually map where the transcription factor is binding across the genome. The rationale being, not all of these changes that we see by RNA-seq are going to be direct effects, but the ones that are, there should be binding of that transcription factor nearby. And of course, the problem that many of us now recognize is if you perform these genome-wide chromatin association studies, here cut and run for using the HA tag on AML1-EDO, we see it binds more than 30,000 places across the genome. And if you were to just annotate those 30,000 peaks very crudely using nearest neighbor, you would think that this transcription fusion has the potential to regulate more than 15,000 genes. But of course, I just showed you that it regulates a very narrow gene signature. And so how do we make sense of the fact that these transcription factors seem to bind all over the place, but they have a much more specific effect on gene expression? And so the way that we set out to do this was to use these direct targets as a roadmap to allow us to focus on what are likely the functional binding events. And so when we do this, we recognize the importance of using a functional filter for enhancer activity. So if we look at all peaks that are bound by AML1-EDO, and we look for changes in H3K27 acetylation, which again is a mark of active enhancers, and we look after AML1 degradation, there's not only is there no change, if anything, there's actually less K27 acetylation globally across AML1 bound regions. But if we focus on the roughly 250 enhancers that annotate to this 59 gene repression signature, now we see when we degrade AML1 there's a significant increase in K27 acetyl. Moreover, for AML1 it's clear that by the time that when you start looking at the more weakly bound sites, and this is just K-means clustering, you can see that there's less of an effect. So it does seem like for AML1 the most functional binding sites are enriched in these upper clusters that have the most robust signal. And so we think it's important when you're trying to define the regulated binding events, you use a functional filter. And I will say, if we quantify enhancer RNA, it's completely consistent with this H3K27 acetyl. It's just these regions that associate with changes in gene regulation where you see these changes in enhancer activity as well. So here's an example of what this looks like. This is the CD82 locus. There's a cluster of intronic enhancers at the CD82 locus, and you see when you degrade AML1 very quickly within 2 hours, you get an increase in H3K27 acetyl across this cluster of enhancers. And you also see an increase in this antisense eRNA transcription as well. And that correlates then with an increase in transcription through the CD82 locus. CD82 is nice because it encodes a cell surface protein, and we can actually easily quantify changes in protein levels by flow cytometry. And you can see when you degrade AML1 there's a dramatic increase in CD82 protein on the cell surface. Okay, so just to quickly summarize this part, I hope that I've convinced you that rapid protein degradation allows identification of direct targets. In the context of AML1 it really seems to repress a very limited number of genes, and the de-repression upon degradation happens quite quickly within 2 hours. So I hope from this part, what I can start to convince you is that binding is not necessarily the same thing as regulation. And this is not something that's specific to AML1 we've seen this really for every transcription factor we've looked at. A number of other groups pairing this Degron technology with genome-wide binding data have come to the same conclusion. And actually this paper just published in Nature from the Han lab was actually quite impressive. They essentially looked at every yeast transcription factor by combining this Degron technology and also genome-wide mapping. And the title of this paper was Low Overlap of Transcription Factor DNA Binding and Regulatory Targets. So as we finished up this AML1 story, this was what struck me the most. How binding really, in the case of AML1 it was maybe 1% of the binding events we could attribute any sort of functional activity to. And so why might this be? Well, here's just some of the obvious things that come to mind when we were thinking about this. Perhaps chromatin context matters. Perhaps the other transacting factors that might be present at the enhancer are contributing to whether or not regulation can occur. That's somewhat related to enhancer grammar. The cell state may play a role, and context likely matters, and binding duration and frequency may dictate. So perhaps low occupancy sites, though I'll talk about this later in the talk, I don't actually think that's the case, maybe less likely to contribute to regulation. But this is where our head was at as we came to the end of this AML1 story. So I briefly want to talk about the first one, chromatin context, because actually as we finished this story on AML1 we did see examples where chromatin context absolutely mattered. And so all the data that I showed you came from an A21 cell line. Lots of mutations. It's actually quite abnormal. It has P53 mutation, KIT mutation. Those are not normal in A21 leukemias. And so we wanted to look at the role of the fusion protein in regulating transcription in a more quote unquote normal setting. And so Jim Malloy and Steve Neimer had shown that you could take human CD34 positive cord blood cells. So CD34, these are enriched in the stem and progenitor cells in hematopoiesis, and you can express the AM1 fusion protein, and it will essentially immortalize these cultures. They're not completely immortalized, but you can culture them for months on end. And so instead of infecting these cells just with the AML1 fusion, we infected them with the Degron tag fusion. And just like the wild type, they effectively immortalize the cells, and now we can degrade the fusion in the absence of other mutations. And so when we did this, we observed that really, AML1 binding in either the 821 cell lines called Kasumi1s or CD34 positive HSPCs were broadly overlapping. There was a high degree of overlap as to where the fusion protein went in both of these contexts. And so we profiled changes in gene expression when we degraded the fusion protein in this setting. Cells are somewhat limited in this setting, so we didn't do PRO-seq, we just did RNA-seq, but there was still a fair amount of overlap in the targets, both in the CD34 pre-leukemic system and in the 821 cell line. There were a number of gene expression changes that we found in the 821 cell line but they were all trending in the same direction in the CD34s, but just didn't quite reach our statistical cutoff. And in fact, if you look at CD82, which we didn't see come out of the CD34s, if you wait and flow the CD34s to longer time points, you can in fact see a significant increase in CD82 protein. But what we were really interested in was this collection of genes that seem to be, and GFI1B and NMYC in particular, were two of the most highly de-repressed genes in the CD34s, and we didn't see them changing in the Kasumi1 cells at all. And we wondered why this might be the case. And when we looked at GFI1B, and the same holds true for NMYC, what we noticed was at baseline, the level of expression in the 821 cell line was much, much lower than in the CD34s. And so we decided to look at polycomb-mediated gene silencing, so establishment of facultative heterochromatin by profiling H3K27 trimethyl. And what we saw was kind of the regions that are marked by this polycomb-mediated repressive mark were very different between the CD34 and the Kasumi1 cells. And in fact, if you looked at the GFI1B locus, while the binding of the fusion protein was the same between the CD34s and the Kasumi1s, in Kasumi1s, the locus was marked by this H3K27 trimethyl, where there were no K27 trimethyl in the CD34s. Contrast that with CBP alpha, which is regulated in both cell lines, and you see in both instances they're free of H3K27 trimethyl. And so we think, I think what I would like you to take away from this part of the talk is that GFI1B was only de-repressed in the pre-leukemic cells, which suggested that AML1 loss was not sufficient to overcome the K27 trimethyl. So the chromatin structure in the context is important for when we're assigning these regulatory versus non-regulatory binding events. And I think this also raises an important point when we're using these Degron technologies to study transcription factors, and is that they're very good at defining whether or not the transcription factors are required for the maintenance of a particular gene signature, but they can't determine whether or not these transcription factors are important for the establishment. And so it is possible in the Kasumi1 system, for instance, that AE was important for initially establishing repression of GFI1B, but then there could be more stable epigenetic changes that come along, like polycomb-mediated silencing, and now the fusion protein is no longer required. Okay, so now I'm going to switch gears with kind of these ideas in mind of limited direct targets. How do we discern whether binding events are regulatory or not? And I'm going to kind of explore some of these ideas a bit more using the wild type RUNX1 protein. So same system that we use to study the fusion protein. We use CRISPR, we integrate this Degron tag. Now you can see we can degrade much more rapidly. So in this case, the wild type RUNX1 is completely gone within 2 hours. We do the same PRO-seq assay to first define the direct targets. And what we immediately realized was that the RUNX1 mediated regulation is much more complex than the regulation by the RUNX1 fusion protein. Now there's clear evidence of both activation and repression. We also see that if you just focus on the genes that are activated and go down following removal of RUNX1, there's very complex patterns of regulation. So some genes change very quickly and they change stably throughout the time course. Others take longer to change, while still others are transient in nature. But definitely the early and stable are where we see the most dramatic changes in gene expression. If we do gene ontology, we can see that there's no enrichment of gene signatures in the genes that are potentially repressed by RUNX1, but the genes that are activated are enriched for only 2 signatures through all time points. And those are platelet activation. And remember, this transcription factor is associated with a familial platelet disorder, as well as genes dictating hematopoietic cell lineage. So we wanted to use so we've used an erythroleukemia cell line here to look at RUNX1 function. And the reason we did that is because this linked to familial platelet disorder. So you can take these cells, and it's a very artificial system, but you can treat them with a 4-ball ester called PMA and actually push them towards a megakaryocyte-like cell fate. So the cells will upregulate two surface proteins called CD41 and CD42. And so we did this along with degradation of RUNX1. And what we saw was that there was a profound block. So the most immature cells are double negative, then they transition to a CD41 single positive state. And then the most differentiated megakaryocyte-like become double positive for these two cell surface markers. And you can see when you degrade RUNX1, there's a profound block. The cells can't move towards this double positive state. So we next wanted to understand how RUNX might be both activating and repressing gene expression. And so we modified our HDR vector and now introduced a flag tag into the endogenous RUNX1 locus instead of the FKBP Degron tag. And so we performed IP mass spec, and the data shows recruitment of a number of co-regulatory complexes. Entire complexes were coming out that were consistent with both gene activation and gene repression. So we could see association with both histone-acetyltransferases like CBP and P300, and also association with HDAC complexes. There was a large association with both the mediator complex and integrator, but there was a profound association of RUNX1 with a number of different chromatin remodeling complexes. But what really caught our eye was the fact that RUNX1 associates with a broad spectrum of other sequence-specific transcription factors. Many of these have been noted before. And in addition to just associating with other sequence-specific translocation or sequence-specific transcription factors, one thing that I'm not going to go into, but was really interesting is that RUNX is involved in roughly a handful of recurrent chromosomal translocations. And 4 of those translocation partners came out of our IP mass spec, suggesting that RUNX1 normally associates with many of the fusion partners that it's translocated to in leukemia. And the 5th one that didn't come out of our mass spec wasn't expressed in this cell line. So just kind of an interesting observation. And so we wondered if the complexity of the RUNX1 gene regulatory signature was related to the fact that there might be locus-specific association of RUNX with these other sequence-specific transcription factors. We've profiled a number of them by cut and run. Indeed, there's a high level of localization, but what I just told you in the first part of my talk was that binding alone is not sufficient to infer regulation. And so while I'm not going to go into it today, we've now added Degron tags to 6 or 7 of these other proteins and are interrogating how their regulatory networks overlap. There's a large degree of overlap with a number of these factors, and some are more likely to antagonize RUNX activity and some more likely to cooperate. But just like with AML1 EDO, there's a very poor correlation between RUNX binding and regulation. So only 14% of these RUNX1 bound regions show evidence of changed gene expression. So we wanted to, again, seek out a functional filter to better characterize these different RUNX binding events. And given the high degree of association with chromatin remodeling complexes, we performed ATAC-seq. And we also made use of the PRO-seq data that we had already generated to now quantify changes in enhancer RNA transcription. And so here's an example, the CSF1 locus. You degrade RUNX1 and transcription is rapidly reduced. There's an upstream clustered enhancer where you see RUNX binding, and you can see very specifically at certain regions, you rapidly lose accessibility when you degrade RUNX1, whereas neighboring accessible regions remain unaffected. And then this correlates with a broad reduction in enhancer RNA transcription. And so we can look at this globally, and we see a number of changes in both enhancer RNA synthesis at early time points following RUNX1 degradation. But we were really surprised to see even more changes in chromatin accessibility following RUNX1 degradation. And the changes in chromatin accessibility tended to be happen faster and be much more stable, whereas the changes in eRNAs were kind of dynamic, like those changes in gene transcription that we had observed with the PRO-seq. One thing that we were surprised to see, actually, is that there was very poor overlap between the regions that showed loss of accessibility or eRNA transcription and RUNX1 binding by cut and run. Now, this is to be expected. We expect a lot of cut and run RUNX1 bound regions not to be affected, but what we were surprised by was we could see a lot of changes in accessibility, for instance, for which we could not see evidence of RUNX binding by cut and run. And this has been observed by others as well, that sometimes you can see these regions where these traditional chromatin association studies fails to detect a significant signal. So we looked more closely at using our ATAC data. So ATAC not only can determine chromatin accessibility and map nucleosome depleted regions, but you can actually perform transcription factor footprinting using this approach. So the TN5 transposase is going to integrate into nucleosome depleted regions, but if there's a transcription factor sitting there, it's going to obstruct the ability of the TN5 to integrate right there. And using this footprinting approach, we found another almost 4,500 regions that predicted that something was sitting there and obstructing of the TN5 integration at a RUNX motif. And in fact, when we plotted the cut and run data around this, we do see an enrichment of signal. It's a very distinct pattern where the signal seems to flank this nucleosome depleted region. So we also asked how much overlap there was between changes in accessibility and changes in eRNA synthesis. It was actually pretty small. So we saw some regions where there was both a change in accessibility and eRNA synthesis, more where there was only a change in eRNA. But the biggest group were regions that were either identified as bound by cut and run or by footprinting that showed a change in accessibility, but didn't show any change in eRNA synthesis. So we wanted to know if there was anything, we performed motif analysis to see if there was anything distinct about these different regions that had these different features of enhancer biology changing or remaining the same when we degraded RUNX. And what was interesting is it was really the changes in chromatin accessibility, really the ATAC changes that were most enriched for RUNX motifs. Whereas the changes in eRNA synthesis, if unaccompanied by a change in chromatin accessibility, were really characterized by some of those RUNX1 associated factor motifs, so the ETS factors and the GATA factors. And we then asked, what's most likely to correlate with changes in gene expression? Does changes in chromatin accessibility correlate, or does changes in eRNA synthesis better correlate? And what we found was really it's only changes in eRNA synthesis that correlate with a change in gene transcription, and there's really no significant correlation between these regions that lose accessibility upon RUNX degradation and changes in gene expression following degradation. So we wanted to know, what are these regions where we see these rapid changes in accessibility, but we're not seeing any effect on gene expression. And so we profiled histone marks that are commonly associated with enhancers. And we see these two clusters where, again, you only see changes in accessibility. They're marked by H3K4 monomethyl, which is enriched in enhancer elements. They're relatively devoid of H3K4 trimethyl, which is, again, consistent with an enhancer element. But unlike these changes in these regions where we see changes in eRNA synthesis, they're relatively devoid of H3K27 acetyl, which is, again, associated with active enhancers. And they're more likely to be marked by H3K27 trimethyl. And so many of these features are considered features of what are often referred to as poised enhancers. And so we wondered if RUNX1 was actually working to establish and maintain enhancer poising. And that's why these regions weren't actually associating with changes in gene expression, because while open, they weren't fully activated yet. And so to kind of address this, we went back to this PMA system where we can induce cell state transition. We can induce the cells to start to differentiate by treating with PMA. And so what we did is we treated with PMA for 1, 2, or 3 days to start to trigger the cells to change. And that at each of those time points, we degraded RUNX for 2 hours and asked, how does chromatin accessibility change? And are there changes in gene targets and enhancer activity by PROTAC? And we really just wanted to ask 2 questions. Do any of these putative poised enhancers become activated with differentiation? And are there changes in the RUNX gene network as you trigger the cells to differentiate? Okay, so by ATAC, we can see that PMA induces broad enhancer changes in chromatin accessibility. So there are clearly regions that are closing with PMA treatment. There are a large number of regions that begin opening with PMA treatment. And we can, again, do our transcription factor footprinting to ask, are there certain motifs that seem to be where the protection is changing with PMA treatment? And when we do this, we see that in the regions that close upon PMA treatment, there's a high enrichment of STAT, RUNX, and AP1 motifs. For STAT and RUNX, there's actually a reduction in protection of those motifs following PMA treatment, suggesting perhaps those factors may no longer be there following PMA treatment. Whereas AP1, the protection stays the same across the time point. And it was very dramatic. The regions that open, there's a huge enrichment of AP1 motifs. While they aren't protected at day 0, by day 1 and 2, there's a high degree of protection, suggesting AP1 is likely sitting at these regions. And this is not very surprising because PMA treatment signals through PKC, which feeds into MAP kinase signaling. And in fact, if you look at the FOS and Jun loci, so FOS, the coding transcript is shown here in blue. You see a rapid upregulation, and the same with Jun, which is shown in this brownish red color. You see a rapid upregulation upon PMA treatment. And so we think what PMA is doing is really changing the repertoire of transacting factors in these cells, substantially upregulating AP1, which is now coming and involved in a lot of these transcriptional changes that are happening. We can also look at changes in the PROTAC active eRNA over time. And so just like the changes in accessibility, we see some regions where enhancers seem to be going off, and then other regions where enhancers are coming on upon PMA treatment. And when we asked whether any of those putative poised enhancers that we had previously defined, where RUNX was required to maintain accessibility, but there wasn't evidence that the enhancer was become activated, excuse me, it's roughly 10% of them. So roughly 10% of these regions become activated with PMA induction. We then wanted to ask, how does the RUNX1 gene network change as you induce differentiation? And this was kind of surprising to me. I expected for there to be, new RUNX1 targets emerging and perhaps others lost. But what we really see is that the most gene expression changes happen prior to PMA treatment. And then as you treat with PMA, the RUNX1 gene signature seems to become much smaller. And we can look at the heat map. And so what we see is, particularly for the downregulated genes, there's only roughly 61 genes, so a much narrower gene network that remains sensitive to RUNX1 depletion following PMA treatment, whereas the vast majority of genes that were previously sensitive are no longer sensitive. And in those genes that are upregulated, you can see that by day 2 and day 3 following PMA, there's really very few genes that become upregulated upon RUNX1 removal. And I do, though, want to point out that it's not that just because the gene expression signature narrows doesn't mean RUNX isn't important anymore. So we can go back to our PMA system, and we treated the cells with PMA for 3 days, which was the latest time point that we looked at changes in gene expression. And so the cells are starting to move first through the CD41 single positive and then towards a double positive state. And at this point, we then degraded RUNX1. And degrading RUNX1 3 days after PMA induced differentiation still results in a profound defect in the further differentiation of the cells. So even though the gene expression signature is narrowed, RUNX1 activity is still doing something really important, even by day 3, that's required to promote differentiation of these cells. And so why are genes that were once sensitive to RUNX1 no longer sensitive to RUNX depletion following PMA treatment? And what we see kind of falls into 2 general models. So the first is exemplified by this IL-1 beta locus, where there's an intronic enhancer. It's sensitive to RUNX depletion, you lose accessibility, and that really seems to be the highly active enhancer driving IL-1 beta expression. And when you degrade RUNX, the eRNA goes down, and that correlates with the reduction in IL-1 beta transcription. However, I'm just showing day 2 for simplicity's sake. When you treat with PMA, first of all, transcription goes way up. This enhancer seems to be, to some degree, less important. And now these 2 downstream enhancers are very robustly active, and they're not sensitive to RUNX removal. So in this case, what seems to be happening is new enhancers are activating, and the reliance on the RUNX regulated enhancer is diminished. There's also examples such as CD82, which, if you recall, I said is regulated by the RUNX1 fusion protein as well. And there's this cluster of intronic enhancers that AE represses and RUNX1 is required to keep active. In the absence of PMA, you see a rapid loss in that antisense eRNA production that correlates with the reduction in transcription. But upon PMA treatment now, you can deplete RUNX1 from these enhancers, and that enhancer stays active. And the gene is no longer sensitive to RUNX1 depletion. So to just summarize the second part, RUNX1 both activates and represses gene expression, and it seems to maintain accessibility at least some likely poised enhancers. By using the PMA treatment to kind of trigger in an isogenic model a change in cell state, we see rewiring of enhancer networks, which also altering pools of transacting factors. So now we have a lot of AP1 in the cell. And so while RUNX continues to regulate a small gene signature in this case, many of the gene targets that were previously sensitive no longer rely on RUNX1 to maintain gene expression. And so if there was one take-home message from this part of the talk, I hope it's that while binding doesn't equal regulation, the context really does matter. And in one cell state, while one binding event may not actually be important to regulate a gene, in a different cell state where different cooperating transcription factors are expressed, where different chromatin contexts, where there's different enhancers that are active, that binding event actually may elicit a regulatory function. And so that's all I have for you today. So I'd just like to acknowledge my lab. Most of the RUNX work was done by a very talented postdoc, Joyita. The AML1 EDO work was done at the end of my postdoc while still at Vanderbilt in the Hebert lab. And I have to thank funding, particularly from the RUNX1 research program and the Edward P. Evans Foundation, as well as NIH. And so thank you all for your attention and for being here, and I'm happy to take any questions.
Thank you so much. That was awesome. If anyone wants to ask a question, feel free to type it into chat or just turn your camera and mic on, and we can just take questions as they come. I think we've got a raised hand. Yeah, Phil, go ahead.
I think super interesting data. I was wondering, I had like several questions, but I was wondering, so first of all, do you think DNA binding, it is important whether transcription factor binds to a certain region of the genome through its own DNA binding domain, or for example, through like associating with transcription, other transcription factors through activating domain? Because you showed that like RUNX1 binds to so many transcription factors. And I was just wondering if you, for example, did the DNA binding mutant, whether you think the results would be similar to completely removing the transcription factor, or whether DNA binding mutant, in that case, binding changes and gene regulation changes would match better.
That's a great question. So we've done complementation studies with DNA binding mutants and we've done it with a number of DNA binding mutants because we used actually patient associated mutations that are associated with the FPD families. And you can actually see a correlation between the degree to which binding is compromised and again, this is a very artificial system of differentiation, the degree to which differentiation is affected. So I do think many of the RUNX targets binding is important. There are clearly some instances, particularly with the genes that that RUNX seems to perhaps function in repression, where we really see no evidence of enrichment of RUNX motifs at all. And there it seems like perhaps binding is not as important. But again, the DNA binding mutants clearly are phenotypic. They clearly are impaired in their ability just in a reporter assay to transactivate. And in these differentiation assays, they're impaired in their ability to induce differentiation. But it's very nuanced.
I think you just mentioned that in one of the hypotheses why not always binding changes equals to gene regulation was that the dwell time of binding matters, and you said that you don't think that it's the case. So I was wondering if you can like think.
Yeah, so okay, so for many of the fusion proteins, for instance, that we've studied. So for the RUNX1 fusion protein, we've also published on a fusion protein called PAX3-FOXO that's involved in pediatric sarcomas. In those cases, and this is not a direct readout of dwell time or binding kinetics. We're doing those assays now in the single molecule tracking system, so I can't really answer it yet. But always, the regulatory binding events were enriched in the regions that had the highest chip seeker cut and run signal. So the very weak signal, which could be, less occupancy, tended to not be enriched in those regulatory binding events. Now, it didn't mean that many of the high chip signal, many of those we couldn't assign regulation to, but still, it was by and large enriched there. In contrast, for RUNX1, most of the regulatory seem to be, if you do k-means clustering of that cut and run, they're actually enriched in the lowest cluster. I don't necessarily understand what that means just yet, which is why, again, now we're doing a lot of the single molecule imaging to try to discern whether or not either not only the binding kinetics, but also we do something called hub analysis where you can see, does it have to revisit a site a certain number of times to elicit regulation? So those are the questions that I think I don't have a good answer for that. It's just one of the hypotheses that has crossed my mind and something that we're trying to address. But I think right now we just don't know. And again, for different factors, the answer may be different.
Very interesting. Thank you.
So thank you, Dr. Stengel, for your presentation. An incredible amount of data. I think it's very compelling. Especially interested in some of the ATAC-seq eRNA and the cut and run data. I know that you presented that there's this lack of overlap in some of the outputs from cut and run and say like accessibility. I was wondering if you have considered the fact that there's a long range effect on the three-dimensional chromatin conformation that results from a change in, say, RUNX1 depletion that might be causing some of that lack of direct overlap that we see in some of those data sets.
Yeah, that's a great question. So we're doing those assays now. We're doing promoter capture micro-C to try to get at just those questions, because that's of course a possibility, that it's really a three-dimensional structure issue where RUNX is binding in one place but eliciting these changes in a different locus. So hopefully we'll have an answer soon, but I don't have a good answer right now.
I think I would like to follow up with you after this. So I'm a research associate in the Dean's lab, and obviously I think some of the 3D imaging that we can do, and we can hopefully get really high resolution views of the chromatin domains and how they're changing. I think that some of the data that you presented, if we follow up after this, I think we can hopefully kind of give an idea or at least a hypothesis on why we're seeing some of the changes based on our understanding of chromatin packing domains and how they might be changing. So I think that it's really interesting, especially with the ATAC-seq and the PRO-seq data sets that you have. If we can dive into those a little deeper and maybe yeah, so I think that there's a lot of really interesting data that I got really excited when I saw it, that we might be able to give you a different perspective in how things might be changing.
That would be fantastic. Yeah, please just shoot me an email and follow up.
Okay.
I'd absolutely love to talk more.
Okay, that sounds great. But again, amazing amount of data. I was trying to just process it as you were presenting it, but thank you so much. Really, really interesting stuff.
Thank you.